Okay, so thanks, Jeremy. It's a pleasure to be here and also to see Jeremy again and to talk to some of you about the interesting work that you've been doing. So I'm a clinician scientist and I work in Cambridge, and half of my time is spent as a kidney doctor. So I principally look after people with kidney transplants, but also some people with autoimmune kidney diseases. And then in that context, I've actually been pretty heavily involved in trying to introduce some new immunosuppressants into transplantation. So we heard earlier how that's been a challenge, but particularly immunosuppressants that are focused on limiting B-cell and antibody responses. And then I also have a basic immunology lab, and in that context, we're mostly focused on understanding antibody effector function. And it's that part that I'm going to talk to you about today. So we've heard a lot today about T-cell responses, but I'm much more interested in antibodies and the way that they're very powerfully able to induce an immune response. And as has been pointed out, the predominance of effective vaccines that we have to date, the ones that work, all induce an antibody response. And trying to manipulate antibodies immunotherapeutically is one of the earliest things that we've tried to do medically. So this is Emil von Behring, who immunized horses with extracts of diphtheria to produce diphtheria toxin, which was then given to individuals with diphtheria, and is sort of one of the first examples of being able to specifically use humoral immunity to protect against an infectious disease. And this is, of course, because antibodies can bind via their variable regions to pathogens and the toxins of pathogens, neutralizing them. But also antibodies, of course, can powerfully induce other effector mechanisms in the immune system. So antibodies can harness innate immunity by activating complement via the classical pathway. But also can bind to cell surface Fc receptors. And in the case of IgG antibodies, these are Fc-gamma receptors. And Fc-gamma receptors are expressed on almost all cells in the immune system, apart from T cells. And so that includes NK cells, neutrophils, macrophages, monocytes, dendritic cells, and B cells. And the importance of controlling the magnitude of antibody effector function, particularly in engaging Fc-gamma receptors, is illustrated by the fact that evolutionarily we have ways of both limiting antibody effector function, so to prevent, for example, autoimmunity or sepsis, but also to enhance antibody effector function if we need that to clear infections. And therapeutically, we might want to do that to enhance vaccine strategies and also anti-tumor responses. And immunology certainly has been much more focused on T cells, but there is a resurgence in interest in antibody effector function. So there have been a couple of very high-profile papers more recently. So one which really caught my eye, which was published in Nature a year or so ago, showed that in a mouse model, and they use a number of tumor models, that if you transferred allogeneic IgG, so this is in humans, this would be the equivalent of giving someone IVIG, intravenous immunoglobulin, and then you induced inflammation in a tumor, that actually IgG would then produce an immune response against that tumor. And this was driven by Fc-gamma receptor expression on the surface of dendritic cells in the tumor. So this is pretty exciting because it suggests that, and perhaps not unsurprisingly, that within our antibody pool, we have antibodies that could recognize tumor antigens and could potentially induce a T-cell response via opsonizing tumor antigens and presenting to DCs if we knew how to harness that and if we could understand why in tumors that doesn't happen. And then also an increased interest in, again, trying to augment antibody responses, so this time in the context of TB, which previously people haven't thought that antibodies play a role in. So the importance of controlling the magnitude of antibody effector function is illustrated, I think, probably best by the way in which we have evolutionarily developed ways of controlling or modulating that. So the rheostats we've developed evolutionarily. So these Fc-gamma receptors come in two forms. They can be activating or inhibitory, and that's, I guess, the first way of controlling the response. So in humans, the activating receptors that bind to IgG immune complexes are listed here, and their effects are controlled by a single inhibitory receptor, FcγR2b. So, for example, on the surface of any given cell, for example, a macrophage, if it's expressing Fc-gamma receptors and it encounters IgG immune complexes, this will initiate a signaling cascade that results in processes like phagocytosis and pro-inflammatory cytokine production. But if you co-engage FcγR2b, that recruits phosphatases, so particularly SHIP and SHIP1, which dephosphorylate kinases and switch off that signaling. So you can see that the balance of expression on any given cell of activating to inhibitory Fc-gamma receptor will decide whether it will respond when it encounters an IgG immune complex. So we know that there are certain cytokines that, say, on macrophages will cause a relative upregulation of activating Fc-gamma receptors. So that means that that cell now, when it encounters an IgG immune complex, has a much lower activation threshold and can easily get activated. In contrast, other cytokines, so on a macrophage, for example, IL-4, result in upregulation of the inhibitory receptor. So now, even if this macrophage can see IgG immune complexes, it may not get activated to inflammation. So you can understand the importance of this in deciding if you have a vaccine response to an infection or to a tumour, or an autoimmune response, that controlling the magnitude of surface expression of these two different types of receptors is important. So the other way in which, again, is reflected evolutionarily, the importance of controlling the magnitude of this response, is the fact that there are naturally occurring polymorphisms in all of these receptors, so both the activating and the inhibitory receptor. And these polymorphisms basically mean in the activating receptors you bind IgG more or less well. And in terms of the inhibitory receptor, one of the best defined polymorphisms determines the magnitude of the inhibitory signaling downstream, and I'll come back to that. But what's interesting, and the reason I was asking Miles earlier about Fc-gamma receptor polymorphisms, is you can see that depending on which set of polymorphisms you have, whether you can bind IgG more or less well, it doesn't matter how much IgG you have, or you might only have a little bit of antigen-specific IgG, but you may be able to mount a very significant response. And we know if we look in different racial groups that the frequency of these polymorphisms significantly differs. So we did one, I guess you can see it as an exemplar of how important the balance of these receptors is by investigating a polymorphism in the inhibitory receptor, FcγR2b, so CD32b. And this polymorphism, so shown here, RS10505001, and it results in a substitution of a threonine for an isoleucine within the transmembrane domain of the receptor. And other groups had shown that this polymorphism is found at increased frequency in patients with lupus, and which is, I guess, the archetypal antibody-mediated autoimmune disease. And we then went on to look at what effect this transmembrane domain polymorphism might have on receptor function, and showed that it causes the inhibitory receptor to be excluded from sphingolipid rafts so that it can no longer signal. So at a single cell level, individuals who are homozygous for that polymorphism behave like FcγR2b knockouts. And they have, at a cellular level in vitro, very exuberant responses to antibodies. And if you look at the FcγR2b knockout mice that Jeff Ravitch made nearly 20 years ago, they also have excessive responses to IgG responses following immunization, and in all antibody-associated autoimmune models like college-induced arthritis or immune complex-induced alveolitis. So that means this excessive immune response can result in autoimmunity, so antibody-mediated autoimmunity. But on the flip side, a piece of work that I did when I was a PhD student was to try and look at the frequency of this polymorphism worldwide. And I mentioned for some of the activating receptors that varies, and for the inhibitory receptor it also varies. So you can see that Caucasians, so only about 1% of Caucasians are TT homozygotes, so basically knockouts. But if you look in other populations, so Southeast Asian and African populations, that frequency is much higher. So we hypothesized that although having such a polymorphism is bad news for antibody-dependent autoimmunity, that it may be beneficial to have more exuberant responses when you're challenged with infection. And so we looked at two models. So we looked at strep pneumoniae, and these are two major killers worldwide. So strep pneumoniae and then malaria. And we showed that in both cases, that if you were TT homozygous, you had better responses. And in fact, using data from the Wellcome Trust Centre in Kilifi, that impacted on survival in malaria. So on that background, the importance of this receptor system, I've also been doing some work at a cellular level, trying to examine the impact of these receptors on immune cell function. And I'm going to tell you a couple of stories, one about dendritic cells. So what was known when I came to this is that if you engage FcγR receptors on the surface of dendritic cells and you engage the activating receptor, you will cause DC maturation. So that is upregulation of MHC class II, co-stimulation molecules, and the ability to stimulate CD4 T cells. If you co-engage FcγRIIb, that inhibits that process. And actually on immature dendritic cells, FcγRIIb is very highly expressed. The idea being that's tolerogenic to stop you responding to the immune complexes that we all have circulating. But there are times when you want your DCs to be matured, and there are these signals, so some of them here, that cause specific upregulation of 2b and the presentation of antigen. But I was interested in the notion that, I guess, a major caveat for activating T cells, so for dendritic cells to activate T cells, is that they've got to be anatomically co-localized. So most of this will happen in secondary lymphoid organs, where the T cells live here centrally, and it's here that the dendritic cells must come from the periphery to present antigen to them. So the question being was, what's the importance of antibody in driving the process of movement of dendritic cells from peripheral tissues through lymphatic tissue so that they now become co-localized with T cells? So to answer this question, what I did initially was a very reductionist system in a murine model, so taking wild-type dendritic cells that express both activating and the inhibitory FcγR receptors, and then take the FcγRIIb knockout dendritic cells. So these being a model of what happens physiologically in response to some of those stimuli, and a model of what might happen if you were able to therapeutically manipulate this for benefit in vaccination and in tumour responses. So we made some bone marrow-derived dendritic cells. So I used an EGFP mouse so that we'd be able to see these, and then stimulated them ex vivo with either OVA or IgG immune complexed OVA, and then transferred them into a black 6 mouse so that we were then able to look at how they migrated from the skin. So we transferred them subcutaneously. We gave them 48 hours to migrate to the draining lymph nodes and then harvested those draining lymph nodes. So if we look by confocal microscopy, so in the odd colours, you can hopefully see there that overstimulated DCs, very few of them will actually get to the draining lymph nodes. In contrast, IgG-stimulated dendritic cells, far more of them will get to the paracortex to co-localize with T cells. And if we compare the response in wild-type versus FcγRIIb knockout, so showing what is possible if we maximally stimulate activating FcγR receptors, you can see that there's an even greater number of dendritic cells. And we were able to quantify that over lots of lymph nodes and mice and show it to be statistically significant. We also did this flow cytometrically and showed something similar. Although I would say this really made me realize the notion that, when we do our flow studies, we have to mash up things, that actually the amount of signal you lose is huge compared to what you see by microscopy, and certainly what you see if you do live imaging by microscopy. So we then did a competition experiment where we put wild-type dendritic cells, and we had them labelled with one cell tracker, and FcγRIIb knockout dendritic cells with another cell tracker, stimulated them with immune complexes, and then mixed them and put them into the same area. So now we can say they have exactly the same anatomical constraints. What's their migration? And what we were able to see was that the FcγRIIb knockout dendritic cells, which are shown here in the green colour, were much better at getting to draining lymph nodes following transfer. So in this artificial system where we're stimulating dendritic cells ex vivo, and they're also bone marrow-derived dendritic cells, where you can say, well, those are not real dendritic cells, but in that, given those caveats, still the stimulation will cause them to migrate. So then, in trying to examine the mechanism a bit more, it's obviously well known that one of the signal systems that dendritic cells use to get from the periphery to draining lymph nodes is expression of the receptor CCR7, which responds to the chemokines CCL19 and CCL21 that are produced by lymphatic endothelial cells, both in the tissues but also within the draining lymph node itself. So with one of my colleagues at NIH, so Karen Pietri-Aronin, we set up a cool system where on a slide we have 3 channels. So we have the middle channel in which you can put dendritic cells that are suspended in a 3D collagen matrix. And then in one of the outside channels you run PBS. It was a flow system. And in the other channel you can run a chemokine. So you basically set up a gradient that these dendritic cells that are in a 3D matrix migrate towards. And this is considerably superior compared to something, say, like a Boyden chamber, which is, in my hands, more or less a random number generator. So if you then image these cells, you can see that the immune complex stimulated dendritic cells are certainly much more migratory than the overstimulated dendritic cells. And if we compare the wild type with the FcγRIIb knockout, so again giving us a model of what happens if you just stimulate activating receptors, they now become much, much more directed. And this gives you a summation of many, many tracks, and we obviously subjected this to statistical analysis and showed it to be significant. Now, the obvious reason for this is that Fc receptor cross-linking will actually directly stimulate upregulation of CCR7. And so we did some surface staining for CCR7, and you can see particularly in the knockouts a significant increase. And then I wanted to go on to look, not obviously being limited to mouse models, but we're more interested in humans. So we took some human monocyte-derived dendritic cells and stimulated those, so these might even be my own. The immune complex stimulated monocyte-derived dendritic cells move much more than the overstimulated towards a gradient of a chemokine. And we did that over a number of individuals. So obviously the one that did the best were my monocytes, not that I'm competitive. And we similarly showed that on human monocyte-derived dendritic cells, immune complex stimulation caused upregulation of CCR7. So one of the other key things that dendritic cells need to do to get from the periphery to a draining lymph node is so they need to be able to respond to the chemokine gradient, but they also need to be able to digest some of the material that comes in their way. And we know, looking at MMP3 and MMP9 knockout mice, that their dendritic cell migration is severely impaired. So this was an obvious system to look at. So the first thing that we did was to just use a simple FITC gelatin assay. So I took some cover slips, coated them with FITC gelatin. You then culture your dendritic cells on top of them and stimulate them with immune complexes. And if they produce a gelatinase, it will munch up some of the FITC gelatin, so you can then quantify the amount of black dots that appear. And you can see, hopefully, obviously, that the immune complex stimulated DCs produce more of these MMPs. We also quantified that in culture supernatants, shown here the mouse data. And then also we did that in the FcγRIIb knockouts at 2 different time points, and then in human monocyte-derived dendritic cells. So we did some other mechanistic work here, which I don't have time to show. So particularly showing that in mouse, this is FcγR3 dependent. So that gives you then a target to go after if you want to either enhance or reduce this response. But all of the work that I've showed you, one could argue that this is in a very derived system. They're all bone marrow-derived dendritic cells, and there's been a lot of debate about what those cells represent recently. And so the question we wanted to answer was, do immune complexes, can they actually stimulate dendritic cell migration in vivo? So to answer this, and this is one of the reasons I went to Ron Germain's lab, so as many of you know, he's the daddy of many things, but it's certainly the daddy of two-photon microscopy. So two-photon microscopy, you use longer wavelength light, and this allows you to go into tissues without cutting them. So the longer wavelength light penetrates deeper and is not as toxic. So what it allows you to do is image anaesthetized animals, and it gives you the fourth dimension, which is time, so you can see what cells are doing over time. So this is a popliteal lymph node and some B cells and T cells buzzing about together. So the system that I used, so I had the CD11C EYFP reporter mice, so you're able to image dendritic cells. And I used a skin stimulation system where I was putting into the footpad either over or immune complexed over. And then imaging the dermal dendritic cells. And so the sort of images you get, so at the surface, you're able to see the Langerhans cells, and then as you go down through, you come to the dermal dendritic cells, many of which are lying next to blood vessels. This is if we were taking a journey on through. And then if you go on to image, and what we did was to immunize a mouse with over in one paw, immune complex in the other. And then you can actually alternate between the two, so do an hour of imaging here, an hour there, and back and forth. So you're able to see that immune complex stimulated dermal DCs move more, and we can quantify that over many movies and show it to be significant. And I crossed the FcγRIIb knockout mice with the CD11C YFP mice, so we could image DCs that lack the inhibitory receptor, and they really migrate far more efficiently. And again, we can quantify that and show it to be statistically significant. So one of the things that the reviewers asked us to do, and we were obviously imaging cells moving there in the skin, but we're not showing that they're actually moving into lymphatic vessels, which is what they need to do. So we took on a technically more difficult thing, which was to do in vivo labeling of the lymphatic vessels. So you actually give a fluorescently labeled anti-live-1 antibody into the footpad, allow it to drain to label up the lymphatic vessels, and then if you leave it an hour, the non-specific stuff goes away and you're left with being able to image the lymphatic vessels. So this is now, we're able to actually see dendritic cells getting up and going in and moving along the lymphatic vessel. So that allowed us to address the in vivo, the very first part of migration, so the getting up out of the skin and into the lymphatic vessel, but it doesn't tell us whether they actually complete their journey. So to do this, we used a modified version of Gwen Randolph's FITSI paint model, where if you paint the skin with FITSI acetone, you label the immune cells. It gives you a snapshot of the immune cells that are there in the skin, so that when you then go to the lymph node, however long afterwards, you can see the ones that were in the skin. And so we used this in combination with either giving over or immune complexed over, and then harvested the lymph nodes 48 hours later. And then we were able to identify the FITSI positive, MHC class 2 positive, CD11C positive cells, and we also looked at subsets 103 and EPCAM. But you're able to see there's a significant increase in cells that actually complete the journey and get to the draining lymph node where they can stimulate T cells if you give immune complexes, and particularly if you remove the effects of the inhibitory receptor. We then got, in response to the reviewer's comment, that not that I'm bitter at all, it made it a much better paper. We then got the CCR7 knockout mice to show that this was CCR7 dependent, and it basically completely abrogates immune complex generated DC migration. We showed that this was a DC intrinsic effect by taking CCR7 knockout dendritic cells, making them ex vivo, stimulating the immune complexes, transferring them, and you can see they just don't get there. So this process is very definitely CCR7 mediated. So then to take it to a disease relevant context, so one of the diseases that certainly as a nephrologist I'm interested in is lupus. So as I said to you, it's the archetypal IgG immune complex mediated autoimmune disease. So these immune complexes become deposited in the skin, so which is why these models are certainly relevant, but also the kidneys, and they're in a proportion of patients they cause kidney failure. So what we did was to take IgG immune complex containing serum, both from a mouse model of lupus, so the NZBWF1 mice. So these were about 6 months old and all had ANA positivity. And then we actually had access to patients with lupus from the clinic. So what we did to start with was to repeat our in vivo imaging, but stimulating with either wild-type heat-inactivated serum or the NZB-WF1 serum, and we were able to see a really significant increase in the dendritic cell migration that was stimulated. And what we then did was to take some of these serum samples, divide them into two, run one through a column where we depleted IgG and then repeated the experiment. And if you take out IgG from the serum, you basically abrogate its ability to stimulate dendritic cell migration. So we then did again, this experiment was also in response to reviewers' comments. So we then did an experiment where we repeated the FcγRIIb paint experiment, but this time we transferred autoimmune-containing serum IP and looked at the subsequent effect on DC migration. So this is, like a patient who has circulating ANAs or antibodies that are specific for tissues. And we then did the FcγRIIb painting and looked at downstream lymph nodes and could see that the mice to which we'd given the autoimmune sera have an increased number of dendritic cells that get there. And then because, of course, human IgG can bind to murine FcγR receptors, we can actually use the human lupus serum and the healthy control heat-inactivated lupus serum, and we're able to show that the same thing was true. And in fact, the extent of migration that was stimulated was higher in patients that had a higher lupus disease activity. So there's a disease activity score called SLEDI. So overall, this really identified a novel function effector function for antibody that actually completely independently of any TLR engagement, that IgG can cause the mobilisation of dendritic cells to the lymph nodes. So in lupus, clearly this can drive an abnormal immune response, and also has implications, as I've said, I think, for vaccination and perhaps even tumour responses. So from there, so after I came back from NIH, I set up my own lab in Cambridge, and we've been looking at two things. So one of them, we've been looking at, I guess, the two sides of this, okay? So a context in which you want to reduce dendritic cell migration, so that is in antibody-mediated autoimmunity. So we've had a collaboration with GSK to use a small molecule inhibitor that they have, and have repeated some of these assays. And I'd love to be able to show you that data, but which is almost to the point of publication. But as I was telling Jeremy, as an academic, you mostly write your talk on the train on the way to the meeting. And if you have a collaboration with some industrial partners, then if you want to show a slide, it requires a 3-month process of approval to show that slide. So I can't show you any of that data. And then the other thing we've been working on is the idea that the expression of FcγR receptors on tumour mononuclear phagocytes, and particularly the idea that the ratio of expression might be important. So as I said, this was based on this paper where they showed that, so if you transfer here IgG, you can cause a significant reduction in tumour size. If you transfer it to an FcγR receptor knockout, you don't see that. And this paper also showed you needed an additional stimulation, and this was dependent on mononuclear phagocytes in the tumour. So this really raised the hypothesis that perhaps the balance of expression of activating to inhibitory receptors in tumour mononuclear phagocytes is actually skewed significantly towards the inhibitory receptor, so thus preventing the tumour from responding. It's only when you give an inflammatory signal that changes. So we've begun to do some work on this in some mouse tumours, and the tumour-associated macrophages in this case are shown here in red, and bone marrow-derived macrophages from the same mouse in blue. If we plot the ratios of the two, we can see that really the tumour-associated macrophages, there's a real drive towards inhibition. And what we want to take this through to using a mouse model. We can obviously use the FcγRIIb knockout mice to say, well, what happens if we're able to neutralise FcγRIIb completely? Does that actually allow IgG to suddenly do away with, or at least reduce the size of some of these tumours? And then alongside that, we've also been working with some clinical colleagues in urology to begin to characterise mononuclear phagocytes in tumours, so using CyTOF. And we've just really started this work to get a reasonable panel together and actually initially looking at tissue-resident cells within kidneys, using kidneys that are donated for transplant but can't be used, which are a rich resource for research. So we hope to be able to take our mouse observation into the human. Okay, and then the much shorter story that I'll tell you is, so I've talked to you about the potential dendritic cell intrinsic impact of FcγR receptor cross-linking. But another thing that we've been thinking about is whether IgG antibodies can actually control the magnitude of the immune highways, so the network of lymphatic vessels that dendritic cells use to travel. And so we've been actually particularly interested at the lymphatic infrastructure of lymph nodes. And this is a confocal tile that I took whilst I was at NIH, showing that, so using Live1 to label up lymphatic vessels and showing that, a normal there's an inguinal lymph node in a mouse. It has a really dense network of lymphatic vessels that can traffic cells to particular areas. And Gwen Randolph showed a little while back now in this immunity paper that during an immunization, so an LPS-type immunization, that there's significant remodeling of that lymphatic infrastructure within a lymph node. So this is unimmunized and then actually CFA-KLH. And she showed that this was dependent on B-cell production of VEGF-A. And so on the basis that these lymphatic vessels, and they also showed that you expand that lymphatic vasculature, you increase the number of dendritic cells that get to the T-cell zone, and also increase subsequent responses. And clearly that's important in a number of contexts, and also that dendritic cells can do a number of other things when they get there. And a number of groups have also shown that lymphatic vessels are important, have other roles, for example, in presenting self-antigen. So I was interested in testing this hypothesis. So can IgG alter that lymphatic infrastructure? So what we did to start with, and to summarize a whole lot of data, so in vitro we stimulated macrophages, dendritic cells, and B cells, both from mice and from humans with immune complexes, and showed that caused the production of VEGF-A, which is what the Gwen Randolph paper has shown was important. So then we wanted to know whether that mattered in vivo, and in particular, many of you will know that within a lymph node, there's a very particular subset of macrophages called the subcapsular sinus macrophages that, as their name suggests, line the subcapsular sinus, and they're basically the first port of call for if you immunize something subcutaneously and it gets drained, they are the cells that pick up immune complexes and anything to start with. So what we then did was set up a system, so the same system used by Jason's sister, where you immunize, where you give an anti-P antibody, passively transfer it, and then you immunize with P to generate a local immune complex. And we then did some confocal imaging, so looking at whether, if you therefore deliver immune complexes directly to the subcapsular sinus macrophages, does it make them produce VEGF-A? And the answer is yes, we were really nicely able to show that. And we're able to do some co-localization. And then move on to asking, therefore, does that, so that's looking at 24 hours, therefore, does that lead on to the generation of new lymphatic vessels? And so we used a slightly different system, but an initial immunization to generate IgG, and then a secondary immunization with OVA, so then you're, again, draining immune complexes into the lymph node. And you can see that does indeed expand the number of lymphatic vessels, and we can quantify that. We were able to show, by using the FcγRIIb knockouts here, that it's dependent, it's controlled by FcγR2b, that this does practically matter. It increases the number of dendritic cells that you drain to those lymph nodes, and that it's dependent on VEGF-A and its interaction with VEGFR2, and we were able to block it with an antibody. We also have mice where we have FcγR2b specifically overexpressed on macrophages, so we're able to use them, and what you see is the converse of the picture in the FcγR2b knockout, so there's less lymphatic vessels and less dendritic cells. So to summarize that piece of work, this shows that IgG antibody can not only alter the ability of the dendritic cell to migrate, but also completely change and expand the the conduits that it's using. And we went some way to looking at the signalling behind that, in that we showed that this was ERK dependent. But some work that we've been doing since then, I mean, I've reflected that the generation of VEGF-A, VEGF-A is best known as I guess one of the things that's produced in a HIF-1 alpha dependent fashion in tumours. And the notion that antibodies and Fc receptors induce HIF-1 alpha is actually not one that's greatly explored or even known. And as an immunologist, you'll know that there's a great deal of interest in the notion that HIF-1 alpha is actually important in determining the metabolism of macrophages. So Luke O'Neill in particular has shown that if you stimulate macrophages with LPS, that in a HIF-1 alpha dependent manner, this switches them to glycolysis. And actually that switch is required for a number of effector functions. So for example, the production of IL-1 beta or ROS. And so we wanted to ask the question that given our data on the VEGF-A, whether actually IgG immune complexes switch cells to glycolysis in a similar manner. And this also raises the notion that you could block this process and that would be or have efficacy in immune complex mediated disease. So what we first did was a transcriptomic study where we took some bone marrow derived macrophages stimulated with immune complexes. And actually this was a little while back, so we did this by microarray, and you can see an upregulation of HIF-1 alpha as well as a number of its target genes. And we also confirmed this at a protein level, this increase in HIF-1 alpha in both normoxic and hypoxic conditions. We then went on to say, well, we can definitely see the upregulation in HIF-1 alpha. What about the effects on glycolysis? So again, using that same data set, we were able to show an increase. So most of you probably won't have the enzymes that are involved in glycolysis in your mind. They're listed here. We could show an upregulation of many of these genes, and in a human data set, an enrichment of glycolysis genes. We then used a seahorse analysis to show whether actually, if we measure extracellular acidification rate, whether immune complex stimulation can cause this switch to glycolysis, and you can see that indeed it does according to that. And that we've got proof of principle that blocking this pathway may be useful as an anti-inflammatory. So using 2DG, which is an inhibitor of glycolysis. So this, I'm just showing you the data from prostaglandin E2 production stimulated by immune complexes, is significantly reduced. So this gives us, I guess, an addition to the current dogma that actually this pathway might be relevant in immune complex mediated diseases and provides a potentially a new pathway for therapeutic targeting. Okay, so those are all the stories I wanted to tell you. So some of the work, the two-photon work I initially did in Ron's lab at NIH, and this is my current lab, and the people in red were involved in some of the studies. So particularly Chen Zi's been doing the glycolysis work, and Gemma doing the inhibitor work that I mentioned. I'm happy to take any questions.